Alright, welcome back. It's good to have you guys once again. So we are going to continue our video series. In the last class we talked about one of the cell divisions, mitosis, right? So I think let's move on to meiosis today. So today our topic for today is on meiosis. So this is Sir Lawrence. I'll be taking you guys this topic today on meiosis. Alright, so I believe you've gone through the last class on the mitosis, so you just have to go through that because in sequence. First of all, you go through the introduction to embryology, then you go through your cell division, mitosis, then we are going to discuss meiosis right now. Then after this class we'll talk about chromosomal abnormalities and then cell cycle before we go into gamete formation, gametogenesis, that is spermatogenesis and oogenesis. Alright, so let's talk about what we have for today, meiosis. Alright, so what's meiosis? That's the first question we are going to ask ourselves right now. What's meiosis? Alright, so meiosis is simply a process where a single matured cell, it must be a matured cell, alright, it must have attained its maximum size, it will have its normal constituent. All cellular constituent must be intact, must be a matured cell, right? Although there are some cases where the cells don't get to maximum size, like the case of labile cells, which we talked about, right, that are going through continuous division. Alright, so let's look at this case where you have a matured cell that divides. This cell will divide twice, not once. In mitosis it divides once, right, and forms two identical daughter cells. But in meiosis there will be division twice, and the result is 4 daughter cells that are non-identical. So it's going to form 4 non-identical daughter cells. Are you with me? So these 4 non-identical daughter cells will contain chromosomes that are half the original number. Like the original number in a somatic cell is 46, right? But at the end of this division, it's going to form cells that have 23 number of chromosomes. First of all, because it has 2 divisions, all right, it has 2 separate divisions, 1 and 2. So after the first, it's going to form 23 paired, 23 bivalent chromatids, all right, which will further divide and form 23 single chromosomes, all right. So this division is going to form 4 non-identical daughter cells containing half the haploid number of chromosomes. Alright, so it's known as the reduction division. Why is it called reduction division? Because it reduces the number of chromosomes, and the reason for that is it usually produces cells, gametes, so that when fertilization occurs, that is going to produce sperm cell with 23 number of chromosomes, and then your matured oocyte with 23 number of chromosomes. So when fertilization occurs, what happens? The diploid number will be restored. That's 46. That's 23 from the sperm, 23 from the matured oocyte. That's 46 chromosomes. Alright, so that's the reason, the importance of this cell division, meiosis. So it's a reduction division, and the purpose is to produce 4 gamete cells at the end. Yes, for sperm formation, it's going to form 4 sperm cells at the end. Alright, it form 4 sperm cells that will exist. You get those sperm cells will still go into further division continuously. Alright, but for the oocyte, matured oocyte, it will go into, first of all, it will form 2 first, then they degenerate. Alright, one will degenerate, then that one will go into another division, the second division, and form another 2. It will degenerate, only one will exist. Will now exist as a matured oocyte. Alright, but in total it's going to form 4 gamete cells. So it involves 2 divisions, and it's important for gametogenesis, gamete formation, that's formation of sperm and the oocyte. That's important. So it has 2 divisions. We said it has 2 divisions. It had meiosis I, and then we have meiosis II. And in meiosis I, you have the interphase 1, prophase 1, metaphase 1, telophase 1, and cytokinesis 1. Then second meiotic division, we have interphase 2, but it's very short. It's very short, and no DNA duplication occurs there. All right, so we have that interphase 2, we have prophase 2, we have metaphase 2, we have anaphase 2 and telophase 2, then cytokinesis 2. So we're going to discuss each of these in details now. So now let's start with the interphase 1. You can see the cell at interphase. Anything interphase is the phase between successive cell division. So it's the phase between the end of one meiotic division and the beginning of another meiotic division. Alright. So at this phase now, the cell is at rest, is getting ready to go into another cell division. So what happens now? You can see here you have your chromosomes here, which are long, thread-like, and they are not visible under a light microscope. Alright. And then you can find your nuclear membrane, and then you find your central, which is a very important organelle that forms spindle fibers during mitosis. Alright. So what happens now? There will be DNA duplication. The reason for this DNA duplication is to double the genetic content. Alright. It will double the genetic content, and then this will result in the chromosome becoming bivalent. It will divide into two chromatids. But these two chromatids that will be formed will not be distant. They will still be joined. They will not be distant. Alright. And this chromosome at this stage will not be visible. Alright. It will still be thread-like, long, and will not be visible under a light microscope. That's what happens at interphase. Alright. Interphase 1. So now what happens next? At prophase 1. Prophase 1 is a very, very long stage. Yes, it's a long phase in meiosis 1. And it comprises of 4 or 5 sub-phases, which are liptotin, zygotin, paccatin, diplotin, and dikinesis. So these are the 5 stages: liptotin, zygotin, paccatin, diplotin, and dikinesis. So let's discuss what happens at each of these stages. So it's just simple. It's just for you to know what happens, and then you'll be able to explain what it is. Just you need some key points. So the thing that happens here, what happens here at this liptotin is just what happens at early prophase in normal mitotic division. The chromosomes are already bivalent, and what causes them to be bivalent is DNA duplication. Alright, so they become bivalent, joined together by the centromere, but they are not distant yet. Alright, they become rod-like. The reason why they are rod-like in appearance is because the chromosome tend to start to coil. It coils more and more, and as it coils, it tends to become turned into a rod-like shape. Alright, so it's because of the coiling of the chromosome. So, but it's not yet visible completely, and not yet distant under a light microscope. Alright. So what happens at zygotin is this: is a process called synapsis. That's the simple thing, the keyword you just need to know: synapsis. So what is the synapsis? You know, in a cell you have 46 number of chromosomes. Alright. So out of this 46 number of chromosomes, we have 44 autosomes, right? And we have 2 sex chromosomes. Alright. So out of those 44 autosomes, we have 22. We have like 2 of them that control a particular characteristic, 2 that control another. Like for example, your color. There are 2 chromosomes in your nucleus that control a particular characteristics, like color, your height. So those ones that are similar, that control a particular characteristics in an individual, they are called homologous chromosomes. Alright. So those homologous chromosomes, like for example your sex, the ones that control your sex is what? X chromosome and the Y if you're a male, or the X and X if you're a female. So they control a particular characteristic, sex. So those homologous chromosomes, what do? They come to line side by side. Alright, in this state, zygotin. So they are going to form what we call the homologous pair of chromosomes. They will form the homologous pair at this stage. And so what is this called? This process is called synapsis. Alright, so look at what I mean here. This liptotin, and you can see the cell not yet distinct. The bivalent chromatids are not yet distinct. You can see the nuclear membrane. Alright, not yet distinct. The central still exists there. Alright, then you can see at zygotin that the chromosomes now that are similar, they have similar characteristics, homologous chromosomes, they come to line side by side. So that's it. Alright, so now let's go into the next stage. We are done with liptotin. We are done with liptotin and zygotin. Let's talk about pacatin. So at pacatin, what happens? This it's homologous pair now, right? The homologous pair. So each of the chromosomes that form the homologous pair, each of them we split, we divide and become bivalent. So this is one chromosome and this another chromosome, alright? These two, they are one homologous pair, alright? So this homologous pair is going to divide. So you can see it has already divided here to form what? Two chromatids, that's bivalent chromatids, that are held together by this centromere. And this another chromosome forming the homologous pair that divides to form the bivalent chromatids. Alright, that's what happens at pacatin. That's the only thing that happens at pacatin. And so in total, you can call this whole chromosomes here tetrads. They are called tetrads in total. Alright, so liptotin is a very, very important stage in garment formation. Why? Because this is where the difference in the different garments form will occur. For example, every sperm formed at the end, they are not similar because of this stage. Every matured oocyte formed at the end, they are not similar because of this liptotin. So what happens at this stage? Look at what happens. Remember, you have this structure here, bivalent chromatids that are divided each into by homologous chromosomes. Each of the chromosomes that are divided into bivalent, right? So what will happen is this: we have this central chromatids, right? This central chromatids, they tend to cross over each other. They form multiple cross over each other, and as they form multiple cross over each other, they have a point of connection. That point of connection is called chiasmata. That you can see, this is one of the point of connection. It's called chiasma here, and many of them is called chiasmata. So this point of connection, what happens now? They become coiled over each other and they form a number of points of connection, right? That point of connection is called chiasmata. So what happens here is this: these chromatids will tend to pull away from each other because the chromosomes now forming the homologous pair, they try to pull away from each other, and they try to pull away from each other, and they have already joined to each other. The central has joined to each other. What happens? There will be a break in the arm. So let's say you can see this red one, right, and this blue one. This blue one will break, the arm will break here, and this red one, the arm breaks here. Alright, so what happens now? Once it breaks, the loose piece is going to join with the opposite. So when this red breaks, it will join with the blue, and when the blue breaks, it joins with the red. That you can see here. So you can see the blue taking the arm of the red and the red taking the arm of the blue. So this process now that occurred here is called crossing over. And the reason for it, the major importance of this is that it helped in exchange of genetic material between these two chromosomes. Alright, so there have been an exchange of genetic material. That's what happened at that stage. So it's called crossing over. Alright, so that's what happens. This is it here. That's it here. And then you have chromosomes that have different characteristics. They are not similar anymore because of this crossing over. So now let's go into dikinesis, which is the last part of prophase 1. So what happens here? There will be a breakdown of nuclear membrane. There will be nucleus will disappear, and then the centron on the opposite poles of the cell start forming spindle fibers, because at this late diplotin, the central have started migrating to the opposite pole of the cell, alright, to the ends of the cell. So central will start forming microtubules that then forms these spindle fibers, alright, that will run from one end of the central to the other. So that's what will happen at this dikinesis. The major thing you need to remember is the breakdown of this nuclear membrane and the nucleus disappearing, alright, and this central forming spindle fibers. So now that's what happens here. You can see it here. The nuclear membrane will break down later, nucleus will disappear, central, they're moving to the opposite poles. So now we'll talk about the next stage. So we've talked about interphase 1, we've talked about prophase 1. So now let's talk about metaphase 1. So at metaphase 1, look at what happens. The central now forms this microtubule that forms this spindle fiber that runs from one end of the central to the other. You can see it running from one end of the central to the other. So what